This method can help answer key questions in cell biology, including unraveling cell signaling pathways. So the main advantage of this technique is that it actually doesn't require any sophisticated microscopic approaches. It's also very fast and therefore usable for real-time observation of protein distribution in cells.
The evaluation and quantification of membrane partitioning of peripherally associated protein is complicated due to light diffraction that causes a mixing of plasma membrane and cytoplasmic fluorescence under the microscope.
Begin this procedure with preparation of biological material as described in the text protocol. Stain the prepared material with FM464 dye by applying a staining protocol that is appropriate for the studied material. For tobacco BY2 cells, stain 200 microliters of cell suspension with 0.2 microliters of 10 millimolar FM464 solution in dimethyl sulfoxide. To capture one equatorial confocal section per one cell, set a sequential two-channel scanning for the chromophore used as the protein tag and for FM464. Also set a higher optical resolution. An example setup is a 63 times oil immersion objective with a numerical aperture of 1.3 and an image size of 1024 by 1024 pixels. Install Fiji ImageJ distribution and the required macro peripheral. In Fiji, select